for being here at these ungodly hours after the lovely dinner we had last night. So then today I'm going to share with you some of the results we have obtained during this HFSP grant, trying to understand how the cells decide where to go. So I would like to start by introducing the team. So then the team is me, Yamuna Krishnan, and Nirgov. And this is a picture of us when we met for the kickoff meeting. So we had a lot of fun, and then we came with very new ideas, actually. So then pretty much like me, Yamuna, and Nir, we have been moving around the world. The cells also move all the time. Cells move in our tissues, and they face constantly directional dilemmas. So what is the first thing that happens? So then they need to decide, they need to sense the microenvironment to see whether there is a preference or a bias. Is there a reason for why going in one direction or not going in another direction? And quite some work has been done to understand that if there is a chemokine, the cells will follow in a certain direction, or if there is an obstacle, the cells will avoid going in that direction. So that this scientific illustration project, in which I co-directed the master thesis of Naomi Wee, you can see a leukocyte. And the leukocyte is going in this direction because there is enough space to go through. Now, if the leukocyte is going in the upper direction, where there is much less space, which we call confinement, now the cell is going to face an obstacle. In immune cells, use the nucleus as the decision factor for deciding whether they should go or not. And as you can see here, the cell faces some consequences. So Naomi put a dramatic touch in the video, as you can see here, but the conclusion remains the same. If the cell takes the wrong decision, it's going to face consequences. But a lot less work has been done into understanding what happens when there is no bias, no apparent bias. So we know that if there is a bias, the cells will take one or the other. But what happens when there is no bias? So for this, how the cells decide where to go has to do a lot with the navigation system of the cells. So the cells polarize and distribute the organelles in an asymmetric way. The cells, and all the cells I will show you in the presentation, normally are moving towards the right. But then, of course, this polarization, which is the asymmetric distribution of organelles thanks to the cytoskeleton, microtubules, and actin, is more complicated than this. So this happens in 3D, actually in 4D. So then in 3D, the organelles are moving. They are spatially distributing all the time. They are contacting each other. So it's a lot more complex that we can see in this cartoon that is very simplified. So we can study directional migration, and cells spontaneously move when we put them in petri dishes like this one, for example. And these cells, you can see, is taking a turn, and it's going now, changing direction from going forward, and now it's going up. But this happens spontaneously. We cannot really control it. So the system we use to control and impose cell shape and then impose a direction of migration is micropatterning. So we use micropatterning, in which we control the adhesion of the surface, and we can make the cells to adhere to only certain places. And this looks like this. So this is a spontaneous migration of cells, and they are only adhering to the red line that you can see here. Well, I hope you saw it at least once. I don't know why, despite checking, it's not running anymore. So then the cells are moving only in these red lines. So then they don't adhere to the gray spaces. So then this is what happens actually in vivo in certain situations. Not all the cells are moving in 3 dimensions. A lot of them, especially cells that are moving within and on top of the vessels, are normally facing only one line, actually. Especially for cells that line on top of the vessels, they experience only what we call lateral confinement. They're moving on top of a line. And this, you can see here, endothelial cells. So then the endothelial cells that are the ones that form the vessels, they spontaneously start sprouting when they form a line on this micropattern, suggesting that actually for these cells, 3D migration is not necessary, but rather 1D migration is enough to reproduce what happens normally in vivo. Of course, this is a simplified model, but this is the logic behind. So then, as I said, this is the idea of the project, what happens, how the cells decide where to go when there is no apparent bias. And this is the case for cells that are moving on top of the vessels, such as macrophages. So macrophages are immune cells that surveil the microenvironment, and they are lining on top of the vessels. And eventually, they will face a junction, as we call them, and they will have to decide between left and right. Same for cancer cells. During metastasis, they co-opt the vessels, and they move within and on top of the vessels to reach different organs. So we use, and we are the cell biologists of the team, so then we use different methods to study the branching and the decisions of the cells. So we use 3D, which is hard to control, and we use microfluidics, including the micropatterns I just showed you. So here you see a macrophage moving in an hexagonal array, facing several decisions at the same time, and you can see that the cell is moving happily in it. So the first thing we needed to have was a quantitative method to understand really how this behavior was going on. So then a brilliant master student in my lab, Jaibai, we discussed about it, and he said, okay, let's check the classical analysis. So then he found the Chol analysis, pretty much using neurons, in which there are drawn concentric circles, and people then count mostly in fixed samples, so they are not moving, count the number of junctions and the number of branches, and they find it like primary, secondary, tertiary, and so on. But considering, you can see the movie on the left, the amount of time points to annotate this by hand, so then Jaibai said, being a Spanish person, said like, no señor, I will find an alternative. And this is when really HFSP somehow happened to be really like the mission of the project and the agency. We received the HFSP Digest, which is this beautiful compilation of all the projects that are running. And I saw this one, which is to study animal behavior. So then I say, well, this is it. I talk to Jaibai, and I say, like, well, you assume, you translate, you use this algorithm, and you translate every extremity as one branch of the cell, and that's it. That was, of course, that was much more simpler in my head than in real life, because extremities are constant in number and length. But as I said, he's a brilliant student, so he managed to re-engineer and change the architecture of the neuronal networks, and he made this analysis in which now we can follow accurately and quantify the branching despite there are new branches forming all the time. Okay, so let's go to the decision making. So here on the left side, you see a cell that is exploring both branches and then is deciding to go left, whereas the cell on the right is going to reach the junction, it's going to explore both branches, but somehow it decides to go back, which we call a reflection. So then first, I want to say that the chances of going to choose one of these three paths is not 1/3. So then the cells prefer a lot more to go forward than to do the reflection. Probably it's more energetically costly. And everything I'm going to show you today is about forward decisions. So cells that are going forward, and then they explore the path, and then decide to eventually decide for one. Along the presentation, you will see several QR codes, which is the link directly to the papers we have published, so feel free to scan them. So here you see another cell that is eventually exploring both branches and deciding to go to the right. Now, if I color code these branches, you can see immediately that the winning arm is always growing, the losing arm grows, and then eventually retracts and shrinks, whereas the cell rear is always shrinking. Now, one thing we already observed, and then everything we did was in parallel to the simulations done by Nir's team, is that there were oscillations. You can see them already as small changes in the length. So then here you can see there are oscillations that emerge immediately. So then now if I take this chymograph, I flip them, so then everything is going to the right just to overlap them. You will see here what happens in the losing versus winning arm at the same time. So at the very beginning, there is equal growing. So then the branches are exploring every path simultaneously and equally. But there is a limitation factor, which is the amount of membrane and the cell length, right? So then after that, they start oscillating, but out of phase. So then for one branch to grow, then necessarily the other one needs to shrink, and then back and forth, making what we call a seesaw oscillation movement. So Nir's team modeled this and found actually that then the model fit very well with making an analogy of every branch being a spring. So for one branch to win, the other one needs to retract, back and forth, until eventually the decision is made and the cells go in a certain direction. But what happened at the moment of the decision? That's the key question, right? So then we went for the usual suspect, and we started by the actin cytoskeleton, which we know that is going to provide the traction for the cells to move forward. And in my lab, we like to compare different cell types, and in this case, we compare endothelial cells, so healthy cells that you can see on top, and cancer metastatic cells, in this case, glioblastoma. The first thing we observe is that the cancer cells, because they are metastatic, they don't really care. They just go forward. They explore very rarely. They just go really fast. So then the healthy cells, on the other hand, they spend much more time in the junction before they take a decision, which made a lot of sense. What happened, despite they have very different speeds of decision, is that we analyzed the actin cytoskeleton, and at the very tip of the cell, so then the very cell front, by developing this other analysis, we checked that even if they have very different speeds of decisions, the reason for which they decide one or the other is the same. And it's that at the moment of the decision, in that oscillation, that branch, when it's growing, it accumulates a little bit more actin at the cell front. So that the very tip of the cell, there is more actin, and that makes that branch the winner. So despite having very different things, there is a convergency in the mechanism, and the winning arm always has a little bit of more actin at the moment of the decision. So this is what happens when the cells are exposed to one decision at a time. But now the next question was, what happens if we put the cells and they face several decisions at the time? So then now here we shrunk the size of the hexagons, and the cells are moving and facing several junctions at the same time. What you can see is that the same cell that was fine doing one decision now is really struggling when it has to decide for several junctions at the same time. So now in this case, we compare endothelial cells, which is what I have been showing you until now, and we compare to macrophages, which are normally used to branch. And as expected, so while the endothelial cell is really struggling here, the macrophage is easily and happily exploring the microenvironment. So Nier's team did the virtual cells, so the simulation for this, and then we can actually model, explain, and actually predict what's going to happen by using this mathematical model. And then we can make the cells by fine-tuning certain parameters that I will not explain for time reasons. We can fine-tune the model to fit like what would be a cell that is struggling with the decisions and a cell that is not struggling with them. We have, in this case, I showed you endothelial cells and macrophages, but we have expanded the library, and we can see that there are clusters of behaviors. So certain cells really struggle, and then you can imagine that they will, and some other cells, such as immune cells that are normally used to branch, they don't struggle with this. But the home message here is that an endothelial cell in the same amount of time, and this is a color-coded time video of the same cell, so then I've just enlarged the macrophage, so this is more or less in a comparable size to the endothelial cell. In the same amount of time, while the endothelial cell explored only once this area, the macrophage already went back and forth a couple of times. But then when we analyze how the decisions were happening in ex vivo, so then ear explants, or in vivo actually, we realized that not always the decision was very symmetrical. Actually, the main reason why we did it symmetrically was to simplify the life of the mathematicians. But then here you see that this, in this junction, it was a rather T-shaped junction. So then this, we assume that that has to do with the potential of the microtubules to bend. So then a symmetrical junction provides 120 angle, then the microtubules will bend only a little. But if we now started changing the angle, this will have consequences in the microtubules. I don't show you the experimental data for time reasons, but then here you see the modeling. Now we are incorporating microtubules in the model, and then here you can see the symmetrical junction, so then the cells is exploring left and right and so on. We have monitored the behavior and the contribution of the centrosome, which is from where the centrosome microtubules emerge. And we have changed the angle to see that actually the harder the angle, the more the cells struggle with the decision. And even in the simulations, you can already see that actually a very sharp angle make in the simulation, also in the experimental data, that the cell actually here decides instead of going forward, it takes a reflection and it goes back where it started. But the 1 million euro, or 1 million Swiss franc, I should say, question is, where are the organelles during this decision? So we start doing simultaneous analysis of organelles in the hexagonal array, but this is extremely difficult. So then in order to move forward, we have to step back. And then we went back to a classical experiment, which is the 2D migration. So the cells will move in a certain direction, and we can analyze. So we use label-free microscopy, as you can see here. And one of the things that appeared was that the mitochondria are going to the front. So then mitochondria are these little stripes that you can see moving to the lamellipodium. To confirm this, we stain mitochondria, as you can see in this video, and actually we observe that in these cells, the mitochondria are really positioned at the cell front where the cell is going, which makes sense because at the cell front, energy will be required, and therefore the mitochondria will be in proximity to provide the energy necessary. Mitochondria, to produce energy, they require very specific control of the membrane potential, so the voltage of the mitochondria. And they do very fascinating stuff. As you can see here, mitochondria undergo fission and fusion, and they do a lot of crazy stuff. And here's where the chemistry comes. So Yamuna had one question, and the question was, are these organelles, so then in this case mitochondria, but the same for other organelles, chemically identical, yes or no? So she developed different chemical probes to interrogate this question, and actually the answer she found is no. Not all mitochondria, they share the same, in this case, pH, which is highly associated to the membrane potential, and they don't have the same one. And actually, there are intra-organelle gradients. So then within an organelle, within a mitochondria, the pH is also different depending on the domain. So then with this, I'm about to finish. So then we check, we distort mitochondrial function, and we saw that actually the cells need it. This was rather expected. And then one of the reasons why this happens is because the cells are detaching. But not all the cells are detaching, because this cell, for example, is detaching, is balling up after we add this drug that distorts mitochondrial function, but many other cells don't detach. So we check for what this drug is doing, and actually is killing the pH gradient between the matrix and the intermitochondrial space. So we used Yamuna's probes, we cut the cell in half, and we evaluated what happened with the pH. And what we found actually is that the cell front, the mitochondria at the cell front are a lot more basic than those at the rear. So we can chemically identify mitochondria and tell where the cell is going. When we use this drug that distorts mitochondrial function, now the gradient is rather killed. So lastly, my last slide is to show you that we went back to the patterns now to make the complex system again, and we put the cells in circles, and then with this radial distribution, and we cut it in half, or in a line, so the cells are going to the right in this case. And we observe, as expected, that the cell in the circles, they have no difference in the pH of the mitochondria because there is no polarity, whereas the cell in the lines, the front, it is more basic than the other ones. So with this, I'm finished, and then I show you today that then during the decision, there are actin-based membrane oscillations, that some cells struggle or can deal better with branching, depending on cell-specific properties, and that we can chemically identify where a cell is going based on in this case, the pH of the mitochondria. With this, I like to thank, and I know that this has been said many times, but I will say it anyways, to the HFSP team, because actually there is no better agency to provide the scientists, what we need. So then an uncomplicated, non-bureaucratic way of getting the money to do risky science, which is no other way to move the frontier of knowledge. I would like to thank my wife for taking care of our little monsters at this moment and allowed me to be here. To the people that actually did the job, so then the people of my lab, and you for your attention. I'll be happy to take any question.
Thank you, Pablo, for a very nice talk.